Awesome. Cool.
Okay. Ready?
I hope so.
Okay, cool. Well, welcome everyone to Emmett Ledden's defense for his PhD. Emmett came borrowed from Truman State, as I was told, and I knew before, and has been working at UNT and also now at UTD via UNT on his PhD. He's done fantastic work, as you will see presently, and has gotten several papers out and many accolades, including GRFP honorable mention, and his poster for this upcoming ACS got selected for GPO. It's not an award. The poster will be judged at the meeting, but just getting selected is a huge thing. So congratulations for that one. Another feather on your cap. And so you'll be presenting it as doctor. Okay, without further ado, here's Emmett.
Okay, thank you very much, Andres. And welcome again to my PhD defense. So let's get started. Okay, that was 1 second. Maybe my slides will work. Okay. As Andres mentioned, I've worked on a number of projects throughout the course of my PhD at UNT. These projects have resulted in 6 publications so far, with 2 more in preparation and submitted. But for today's talk, I'll really be focused on 2 different subsets of projects. So my work with TET2 and my work with POT1. So the first project I'm going to be talking about is about TET2 selectivity and activity with DNA and RNA substrates. The second project will be on different variants within the TET2 structure, within the complex. And then additionally, I'll also be talking about different variants within the POT1 ssDNA structure. So really all of these projects are bound together by the question, how does a different residue affect the complex? How does a different RNA base, how does a mutation, how does something change how a protein complex works? And so we're going to start with TET2. But before we get to TET2, we really have to talk about the central dogma of biology. And the central dogma of biology is known as DNA makes RNA makes protein. Within the genetic code of your body, of life, really, there are different nucleobases. And so those are adenine, guanine, thymine, and cytosine. Adenine pairs with thymine, guanine pairs with cytosine. And this DNA then gets transcribed into RNA. And when you have RNA, that thymine now becomes a uracil. And from this RNA sequence, you can now translate it into a protein where there are 3 amino acids generally will code for an amino acid. And so that's how you get from these different nucleobases to this full protein. And we'll talk a little bit later how different changes within the DNA itself lead to other things. Now, the nucleic acid sequences within your body also are affected by different things. And sometimes that will change the sequence, sometimes it doesn't. When we're talking about epigenetics, epigenetics is a chemical change that will alter the DNA structure, but it doesn't affect the sequence. So usually when I'm going to be talking about stuff here, we're going to be talking about different methyl groups. And methyltransferases are what methylate both DNA and RNA. The most common forms of methylated DNA and RNA are 5-methylcytosine for DNA and N6-methyladenosine for RNA. And approximately 1% of all genomic human nucleic acids are this 5-methylcytosine residue. And the way that this changes is, again, it's just a single methyl group at the 5' position on cytosine. And the reason why these things are important is because these methyl groups are a response basically to your environment. So for instance, methyl groups are a way to silently or not so silently show a change. And so, for example, they can turn on or off different genes throughout time. So sometimes when people are born with blonde hair, they'll later develop darker hair throughout life. And that's as a result of these changing methylation patterns. These changing methylation patterns happen normally throughout puberty and throughout the rest of the aging process. And the reason why I'm spending so much time talking about the single methyl group is because TETs are actually demethylating these DNA and RNA bases. They're part of the iron 2 and alpha ketoglutarate family of dioxygenases, and they act on a flipped in nucleobase. So it's rotated out of the double helix and into this active site where it coordinates with the metal, and it requires alpha ketoglutarate to perform this reaction where they demethylate or oxidize actually this 5-methylcytosine residue. Now, TET2 specifically is important because it's implicated in leukemia and a number of other myeloproliferative disorders, which means that disorders that are within the blood. And somatic TET2 mutations are seen in roughly 15% of all myeloid cancers. And so, like I said, it oxidizes 5-methylcytosine to 5-hydroxymethylcytosine to 5-formylcytosine to 5-carboxylcytosine. So now that we've talked a little bit of what TET2 does, we're going to talk about what some of our experimental collaborators saw. And our experimental collaborators for this specific project was the Coley group at UPenn, and they measured TET2 activity and selectivity. And they found an interesting pattern that they wanted us to investigate further. And so of some of the stuff that they studied, they found that single-stranded 5-methylribocytosine, so a RNA base, single-stranded, was the least reactive. Then double-stranded 5-methylribocytosine, so a double-stranded with RNA. Then single-stranded 5-methyl deoxycytosine, and so DNA and RNA are only different within this oxygen at the 2' hydroxyl. So it's literally one oxygen that is the difference between these two. But for all of the residues in the strand. And then after the single-stranded deoxy, it was double-stranded 5-methyl deoxycytosine. And so the question really became, first, why single-stranded better than double-stranded? Or sorry, why is double-stranded better than single-stranded? And then why is deoxy more favorable than ribo? So why is this double-stranded 5-methyl deoxycytosine the most preferential for activity? And so structurally, we answered this, or tried to answer this, with classical molecular dynamics simulations. And molecular dynamics simulations calculate the movement of atoms in real life using a simplified energy function, and that's known as a force field. And so what's happening is you have a bunch of positions based off of a crystal structure, and you can calculate the force on those atoms at each position based off of where all of the other atoms are using Newton's laws. And so this is done over a different time scale. So typically we're thinking in either the 1 femtosecond or the 2 femtosecond time scale. And you initially give the system a push, and then you kind of see the results of that at every time step thereafter, updating position and velocity as you go. And so with these systems, we studied 9 different configurations for the nucleic acid sequence within the TET2 complex. All of these were with DNA, but the flipped in base was what we would change to an RNA base to study the RNA systems. We used amber, and we used a magnesium 2+ surrogate for iron for the molecular dynamics. And we got all of the parameters based off of a combination of amber and the rest fitting with the red server. And so again, we studied the different oxidation states, as well as both ribo and deoxy for each of these, changing the flipped in base or the complementary base. And so before we get to some of the results that we saw, I want to tell you a little bit about how helical structures change. So when people think about DNA, they typically think about B-form DNA, this nice, clean double helix structure. But there are other forms of helical structures for DNA, and it depends on different conditions, and it also depends on sugar puckering. So when you have B-form DNA, you have what's known as C2' endo puckering or S puckering, and this is really that puckering change, and it has a syn nucleobase configuration. When you go to A form, which is what is seen in aDNA and RNA, you have the C3' endo puckering here, and it changes here at C3', and it has an anti-configuration for the nucleobase. And what these actually end up doing is they change different helical properties. And so things like helical diameter change, things like rise, how much distance there is between 2 different base pairs there are, and how many base pairs there are per turn, which is why you see A form is a little bit more compact than B form. And so really one of the first things that we noticed when we were analyzing these results is if you overlaid the final frames from the deoxy and the ribo systems, this ribo system where again, all it is just one ribo base here in a DNA nucleic acid, you see that it starts to become more compact. And we were curious as to why that was. And so at this point, we started looking at different backbone characteristics. And so we started this search with different delta and chi angles. And when we were talking about single-stranded DNA and double-stranded RNA with the flipped-in base, we saw that the flipped-in base specifically had different populations for its delta and chi angles for the ribo, which is orange, and the deoxy, which is blue. So here for the double-stranded DNA sequences, we saw that everything was a little bit more compact for the 5-MDC base. And again, completely separate from the 5-MRC. And a similar thing was seen with the ssDNA where they're separate, but ssDNA had a lot more, there were a lot more choices basically for it to have a position. It wasn't as tight of a spread on where those angles were. And so we carried out this analysis for the other oxidation states. So this is the 5-MDC, 5-HMDC, 5-FDC, and 5-CAC. All those are blue, and then the ribo again is orange. And interestingly, it's really this 5-MDC that's different from everything else. And we think that actually that's part of why we see what we see with the ribo is 5-MRC actually resembles this 5-methyl or 5-FDC. And because it resembles this 5-FDC, which can form its own form of DNA, F-form DNA, we think that plays a role kind of in why things are different between these deoxy and ribo systems. So to summarize, this 5-MDC is separate, but these 5-MRC are actually resembling these 5-FC systems. And so this is what they are for everything overlaid together, just to show kind of where those spreads are. Deoxy is definitely different from all of the ribo systems and all of the additional oxidation states that we tested. And then for the systems with a complementary strand that had a different nucleic acid residue, these are what we saw. This is that same double-stranded 5-MDC versus 5-MRC. This is one with both ribo here and ribo here in that complementary strand. This one is the deoxy 5-MDC here and a regular deoxycytosine here in the complementary strand. And then finally in green, we have the 5-MDC and then a 5-MRC in the complementary strand where it flipped. And all of the complementary strand systems also were a little bit more spread out than they were in just the 5-MDC system. They weren't as tight. There was a lot more flexibility based off of what complementary strand there was. And so this, again, summarizes all of those changes where all of those things are. And what we really found overall was that both the substrate and the complement are affecting the substrate's chi and delta angles. Now, chi and delta angles are not the only thing that we looked at when we were talking about the backbones. We also looked at two different base pair based ones, shift and slide. So shift is how two different base pairs change along the X plane. Slide is how two different base pairs change along the Y plane. Particularly the two base pairs that we were looking at here were the 3 and 4 position base pairs. And again, we see that the ribo has a lot more of a range than the tight deoxy systems. And particularly when we're looking at shift and slide, you should expect these to be at about 0, 0 for everything. And we see that the ribo kind of leaves that 0, 0 range more than the deoxy system does. So we see this again when you look at the other oxidation states is different spreads. There's a little bit more spread to the deoxy systems. They're not as tight at 0, 0, but overall they're not as dramatically shifted as those ribo systems are at each of the different states. Okay. And so finally, the last analysis I want to talk about for this project is an energy decomposition analysis. And what this was we were looking at different pairwise interaction energies. So one residue with every other residue. In particular, the one residue that we were looking at with reference to everything else was this flipped in base, either the deoxy or the ribo. And what we found was that there were differences throughout the complex at the 1 kilocalorie per mole range with respect to that residue and its non-bonded interactions, so van der Waals and Coulombs. One thing to note here is that we see that there's definitely some shifts for ssDNA that are a little bit, it seems a little bit more dramatic, a little bit more wild type favored than it would be in the double-stranded DNA. And part of that is we believe is due to the flexibility inherent in single-stranded DNA. Single-stranded DNA is a bit more floppy than double-stranded DNA. And the other thing that I want to highlight here is 2 residues that are in the DNA interacting loops of TET2. And so this is one of the DNA interacting loops. This is where R1261 is. And this is a second DNA interacting loop. This is where a K1299 are. Both of those appear in almost every analysis I've ever done with TET2. They seem really important to almost everything and change a lot in their interaction energies for any type of non-double-stranded 5M DC system. Similarly, we see these changes throughout the entire complex for all of the other oxidation states. So that is the TET2 selectivity project. And now we will be moving on to the TET2 variants project. And so earlier I mentioned that sometimes DNA gets changed, and DNA gets changed by a lot of different things. It can get damaged by a lot of different things. So things like sunlight can damage DNA. And sometimes polymerases honestly just have a bad day when they're trying to replicate DNA. And so one of the things that happens when polymerases are having a bad day is that they end up putting the wrong nucleotide in, and you end up with a base mismatch. What was originally what was supposed to be there is not there. Was having a bad day, and that's understandable. But when DNA or when polymerases have a bad day, that can lead to, or that actually does, that's what leads to several mutations. And so there are different types of mutations, all stemming from that change in a nucleotide. But really what we want to focus on are missense mutations, which change a different, from the DNA sequence, changes the RNA sequence, and then the RNA codes for a different amino acid. And so there's different types of missense mutations. Some are conservative, some are non-conservative. We've investigated some of both. And in that particular case, and a conservative codes for something that's similar to what the original amino acid was, and non-conservative codes for something different. So if it was polar, now it might be going non-polar. And so we're interested in specifically what these missense mutations could mean in a TET2 complex. And so this also tries to answer how cancer and mutations are interrelated. So our collaborators, the Iafontis group at NYU Langone, probed the catalog of somatic mutations in cancer for different mutations in TET2. And they chose a few that they thought were interesting. And this led to some experiments being done by the Iafontis and Coley groups, and then simulations being done by our group. And for these, we used a very similar methodology. We investigated 6 different mutations within the TET2 DNA complex. In this particular case, with the 5-HMC substrate. These mutations are throughout the complex. So we looked at C1271S, R1261C and R1261H, I1873T, R1359H, and H1881R, which is interesting because H1881R actually is complexed with the divalent metal cation. And so again, the other thing is when we talk about these mutations, this numbering scheme is, it starts with the first amino acid, and then it's the position where it is within the protein, and that, or actually within the genome, and then the new amino acid is the result. So. But before we get to what we saw, again, what are you going to see? So one of the analyses that we did was a normal mode analysis. And a normal mode analysis really attempts to figure out where the largest contributor to the overall motion comes from. And so the idea is that the ones with the lowest frequencies are the highest contributor. And typically for a normal mode analysis, we're looking at alpha carbons to try and, really extract out what the most important movement is to the overall protein structure. But you can really think of normal mode analysis like the act of you breathing. And so when you breathe, your mouth opens, your lungs inflate, your diaphragm moves down, but your left toe isn't really doing anything. And so if you performed basically a normal mode analysis on you breathing, you should see that the lungs are the largest overall contributor to this motion. And so that's the goal here with normal mode analysis, is to try and find out what is the most important part of that motion. And so with these different variants, we identified 3 different categories of variants. So there were some variants that we saw that affected sites away from the active site, or distal sites. And that one was in particular R1261H. So this first image here is looking at differences in hydrogen bond interactions. Those that are pink are favored in wild type. Those that are orange kind of flipped between wild type and variant, changed in its hydrogen bond interactions. And then those in blue are variant favored. And then in black and circled is the mutation site. And we see that there are differences throughout the complex in hydrogen bond interactions, but also there are definitely some differences that are large away from this active site here. The second one here is the results of the normal mode analysis. And what we saw with particularly this variant was that there was some motion more on this left side away from the DNA, where things were moving around a little bit more than they were in some of the other variants. And then again, we looked at an energy decomposition analysis where we saw some of the large changes away from the active site. There were some changes directly relating to the residue that was mutated, but the largest differences were away from the active site. So now that we've talked about the distal site, there were also active site and comprehensive. So first, we're going to talk about the active site. Those that changed the active site were C1271S and R1359H. Again, there are differences in hydrogen bonding throughout the complex, with some of the most important ones being with active site residues. The normal modes look different than they did in the distal. There's just a lot of things happening around in where this active site is for both of these. There are some changes farther away, but they're in more flexible regions like this brown region, which is where the flexible linker is for this system. And again, results of energy decomposition analysis, there are a lot of large differences within the active site specifically for these residues. And then of the comprehensive, there, again, we saw I1873T, R1261C, both had differences in their hydrogen bond interactions. There were a lot of differences that were a lot larger than they were in some of the other systems. We see, again, that there's just a lot of motion overall for these in the normal modes, independent of kind of where these mutation points are. And changes throughout the system, some centered in the active site and some farther away from the active site for each of these. Now, I did mention separately H1881R. I didn't forget about it. It was also a comprehensive change. H1881R had differences in hydrogen bond interactions throughout the complex, had a similar motion throughout everything within the complex in the primary first normal mode, and then differences throughout the complex. Again, a lot happening within the active site, but several with large differences throughout the complex as well in the energy decomposition analysis. And H1881R was one that our collaborators were very interested in, and that they found was that this variant specifically is inactive. It shows no 5-HMC conversion in vivo, but it does get expressed in mouse models. And in these mouse models, the variant system showed decreased levels of 5-HMC, and that in mice that aged, roughly 90% of those aged mice developed myeloid malignancies at some point. And so we correctly validated that there was some differences within that H1881R system, that because it interacts the whole complex, it's probably leading to some of the things that they're seeing experimentally. So overall, for TET2, we found that the ribo changes the nucleic acid backbone characteristics, that the complementary strand matters, and that variant effects are not necessarily location dependent, and they can have effects throughout the complex or limited to a specific region. In the future, we're interested in the QMM reaction for the ribonucleotides to see if that changes the reaction at all. and can continue working with experimental collaborators to characterize these variants. So now we're going to move on to POT1 and look at different variants within that. So in chromosomes, at the ends of chromosomes are telomeres, and telomeres have a lot of DNA, and they have what's known as a telomeric DNA repeat of TTA, GGG, TTAG. And there are 6 proteins that make up the sheltering complex that are interacting with this telomeric DNA site. And so of these 6 proteins, we are actually really interested in POT1, which actively interacts with the DNA. Not all of these interact directly with the DNA. So POT1 interacts specifically with TPP1 and the DNA itself. TPP1 does not interact with the DNA. And then some of these other ones interact a little bit, or they can recognize it, but they're not directly complexed with DNA. So POT1 is protection of telomeres 1. It serves to protect this telomeric DNA. Now, if you lose this 6 protein complex, it leads to aging and apoptosis. But also, to some extent, you want telomeres to be able to degrade, because at each replicative cycle, telomeres get shorter. And if they do not degrade, if they are starting to be maintained more actively, that type of behavior where it's more maintained is increased in cancer. And so our collaborators, the Robles-Espinosa and Adams groups, identified 37 different missense POT1 variants. And they came to us and they said, we are interested in these 3. Can you tell us what you see? We're not going to tell you what we see, but look into these 3, tell us what's up. And so that's what we did. We studied the 4 different systems. Again, with Amber, these 3 variants are D224N in OB2, which is the oligonucleotide or oligosaccharide binding domain 2. C59Y and K39N are both in OB1, and the ssDNA here is in yellow. And so the very first analysis that we did to look into these 3 variants was, again, the energy decomposition analysis. This time we are looking with respect to the DG6 residue, which is colored black, because DG6 is kind of at like a breaking point between which domain is recognizing which part of the DNA sequence. And so we were interested specifically in how the protein is changing in its interaction with the DNA. And what we found was that all 3 variants had large changes throughout the entire complex. It wasn't just one of the variants that had a change. All 3 of them changed pretty significantly with its interaction with this DG6 residue. We also looked at hydrogen bond analysis, where we saw differences in hydrogen bond interactions, again, along the face of the protein surface, but also some within farther away from the protein DNA interface as well, for each of these 3 variants. But in my personal opinion, the most interesting of these differences was in the normal mode analysis, which sometimes the slide takes a little bit to load. We found differences in both the first and second overall motion for this protein complex for each of the variants. And so wild type, about 60% of the wild type motion is actually coming from like a breathing motion where the 2 domains are pulling apart and coming back together. And its second motion, which is about 20% or 25% of its overall motion, is a rocking of the whole complex. But when you look at the variants, roughly 70 to 80% of their overall motion is not the breathing, it's actually the rocking. And now roughly 8 to 20% instead of rocking is breathing. And so these 2 actually flip their priority for wild type and each of the variants. And so later we talked with our collaborators. They said that all 3 of these interrupt in some form the protein DNA complex that they had seen. And so really, again, we found a similar thing that these 3 variants all affect the protein DNA interactions, and that there's these contrasting overall motions for everything. And in the future, we would like to investigate POT1 TPP's complexing, or TPP1's complexing, because POT1 is supposed to bind to the telomerase DNA first and then bind to TPP1, but existing structures show POT1 and the DNA or POT1 and TPP1. There's not a structure with all three. Similarly, there's not a structure for the entire human sheltering complex. There's some for yeast, but the proteins are a little bit different. And so it'd be really interesting to see how mutations affect the larger complex as a whole, not just this individual protein DNA interaction. And so with that, I would like to thank everyone that worked with me throughout all these projects. I worked, the TET2 MD simulations were a group effort with Dr. Hediye Torre-Bifard, Dr. Pavel Silvestrov, Peter Camacho, Katie Shea, and Corey Shen. The TET2 selectivity and mutant experiments were by the Coley group at UPenn. The H1881R experiments were by the Iafontis group at NYU Langone. I would like to acknowledge Mauricio de Matilde La Garza, who worked on the POT1 MD simulations with me, and our experimental collaborators, the Robles Espinosa and Adams groups. I would like to thank my committee for being here today and for all of their advice over the past few years. And I would like to thank my family and several of my lab mates, specifically Dr. Alice Walker, Dr. Mark Hicks, and Madison Berger for all of the things that they have helped me with over the past few years. And with that, I will take any questions. I will warn you that the YouTube is at about a minute delay. So if you ask questions on YouTube, it might take a bit, but I'll get to those later. So thank you.
Thank you very much, Emmett. That was very nice. And lots of clapping. Thanks. Okay, so at this point, the floor is open for questions from the audience. If anyone has a question, please raise your hand.
Tom,
I think Tom beat Bill. Sorry, so we'll let Tom ask first. Go ahead, Tom.
All right, thank you. Excellent talk, Emmett. I think I've learned more about biology than I have in 5 years. So my question is about the TET2 project or projects. So you had mentioned that you used a magnesium ion in place of iron.
Yep.
I guess why? It does like the spin state matter? Is the iron high spin, low spin?
So yes, we were doing MD simulations, and in MD, that particular all things are treated as point charges in MD, and so while yes, iron and magnesium have different radii, this had actually been tested by Dr. Torre-Bifard to look at both of these, and it was found that the magnesium was a fair surrogate in place of iron. But if you were doing QMMM or if you were doing anything with a quantum region, yes, you would change it to iron. It wouldn't be a fair choice to have magnesium there. But because it's a point charge, you can get away with it.
Okay, and then I actually have a second question of the same thing. So I imagine these proteins in the body being surrounded by water and whatever substrates are in the cells. Does that have any effect on your calculations? Do you do like explicit or implicit solvent models or anything like that?
Yeah, so all of these were done in TIP3P explicit water. And then there's typically some ions to balance charge. So typically we use potassium ions and chloride ions to get to a neutral overall charge for this system. And then similarly, all of these use the particle mesh Ewald method, which is basically kind of like the Pac-Man image where it ends and continues on, and you ideally will have a larger representation. Because of that, it's not just kind of a frozen box, it's ideally continuing.
Okay, and so those ions and solutions and solvents, they don't strongly interact with, like, I don't know, anion pi interactions or anything like that with the proteins? Or
I mean, things can interact, but typically that's not what we're looking at.
You wouldn't expect them to?
Yeah.
Okay, perfect. Thank you.
Bill?
All right. Thanks, Emmett. That was fantastic, as I would expect. I would expect nothing less from you. I actually have two questions, but thanks to Tom, it's only like 1.5 questions.
Okay.
My first question was on that magnesium iron difference. And so my follow-up to what Tom said is based off the technical issues, because it looked like from when you talked about the QMMM, you showed the mechanism, and it looked like that iron was 6 coordinate and sort of an octahedral geometry sort of thing, if that's correct. Did you have to run an mcpb.py calculation to do that, to get that coordination set?
So I did not set up the initial TET2 structure, and I know that there was not MCPBY, all of that stuff for any of these. But so the geometry like is obviously like evaluated before it goes into QMM. The QMM stuff was separate, and I haven't done QMM with TET2. Someone else did. The structure, as far as I know, that wasn't necessarily something. It was based off of that crystal structure of 4NM, like the 4NM6 PDB. I don't necessarily know. I would think that it was, made sure that it was relevant, right, to how iron is, but because like sometimes the water comes in or out, and I don't know.
Okay,
it's all described in the paper, and there was a bunch of energy decomposition analysis, QM, EDA, and all that stuff to make sure. And there are other crystal structures that have the full complex with all of the co-substrates, and so that was all rigorously tested. So.
Okay.
So my follow-up question then, I guess this isn't a follow-up, this is separate, is on your decomposition analysis. On that first project, you had a table of arginine, had an arginine and a potassium in it, or arginine and a lysine in it. Some of the values were very positive, and some of them were one of them was very negative, and three were positive. Could you comment on that? What are they interacting with? Is that the phosphate? Is that the base? What are they interacting with?
This specific, these specific energies are with respect to that flipped in substrate base. So yes, there is some other stuff that can kind of get into that, but ideally it's like, which is why when you do this, you cut out the two directly next to the flipped in base and zero those out. But sometimes, so like with SSDNA, it could just be that it got to a really weird place in one and it didn't get to that really weird place in the other, because again, it's floppy. But whether it's positive or negative, so most of the time I've usually seen K1299N be negative, but it's with respect to which one is favored. And so this case, it's the deoxy system that is favored or the ribose system that is favored. If it's negative, the deoxy system is favored. If it's positive, the ribose system is favored interaction. I don't think I answered your question. I'm sorry.
I think that mostly answers what I was asking. Yeah. Okay, cool. That's all my questions. Thank you. Good job, Emmett.
Thanks.
Thanks, Bill. There's one question from the live stream.
Okay.
It says, do you have an idea yet about how the difference in that rocking versus breeding populations in wild type versus mutants could be affecting the POT1 biological functions?
My personal idea, which is just me talking, right, is that if there is a different motion and it's like it is supposed to act a specific way, if it's now suddenly acting a different way most of the time, then that's going to lead to some issue. And so I think that this specifically would have some sort of effect on its actual ability to be complexed with the DNA, because it's changing its overall motion. It's changing how that interaction is overall.
Cool, thank you. Are there any other questions from the audience? Okay, if there are no more questions, let's thank Emmett again for a very nice talk. And this will conclude the open part of the defense. And so thanks everyone for joining in. And we ask everyone to now please disconnect, and it's the turn of the committee to chat with Emmett. Thanks everyone.